we're going to be drawing it starting from the top left, and we're going to go all the way to the right here. Okay, so just plan accordingly as you're drawing it. Okay, here is a cell. Okay, and we're going to be drawing like a eukaryotic cell. Okay, here is a eukaryotic cell. Okay, or eukaryote. And basically, I am going to zoom in and see what it's gonna be like. Okay, I'm gonna zoom in. There we go. All right. Let's start with the nucleus. So here is the nucleus. Okay, nucleus. Now the nucleus is actually surrounded by a membrane. Right, so we're gonna draw the membrane here together. Okay, and I'm not gonna color the whole thing because I actually want to take a look at the inside of the nucleus in a bit. But here you have it. Okay, I'm just gonna draw maybe 3/4 of it, and then I'm gonna color it in. Hey, hopefully you have some colors with you as well. So there you go. So this membrane that goes around the nucleus, we call that the nuclear envelope. Right? Nuclear envelope surrounds the nucleus. Now, on the nuclear envelope, you will have some openings, some pores. So these pores, they are called nuclear pores. And the nuclear pores are actually useful for controlling what goes into and out of the nucleus. Okay, allows material, allow material to move into or out of the nucleus. Okay, and I'll tell you what kind of things move into and out of the nucleus in a bit. Are you guys able to follow? Do I need to slow down a little bit? Give me some feedback here. Like, are you okay? Yeah, got a thumbs up from one person. Okay, all right, thanks. Let me know, okay, if I'm going too fast. Online it's hard for me because I can't see people's face and I can't see what they're doing. So I'm just trying my best to make sure that it's okay for you guys. All right, now inside the nucleus, there is another circular structure. There's another circular structure right here. Okay, so the whole thing is the nucleus. Maybe I'll just put it right here. But the structure right here in the inside the nucleus is called the nucleolus. Nucleolus. And the nucleolus is important because it synthesizes an other organelle called ribosomes. Okay, we're going to get to that in a second, which is never mind what it does. at the moment, but that's what it does. The nucleolus makes ribosomes. Okay. So what else does the nucleus do? Well, the nucleus also protects genetic material. Okay, so there's some genetic material here, and this is what we call chromatin. I will explain. Okay, chromatin. Chromatin is basically DNA plus, you can say DNA packaged, wrapped around proteins. Now on the side here, I want to explain a little bit more about this chromatin concept. All right, so our genetic material is DNA. And I hope you have been studying because your quiz is on Thursday, and DNA is part of it. And the DNA, as you know, is a double helix, right? So it looks like this. Double helix, and the base pairs are like the ACTGs, right? So DNA on its own, we say naked. It doesn't have anything. Naked DNA. That's actually what it's called, okay? It's kind of rare to find naked DNA flying around in the cell. Instead, what happens is the DNA, they will wrap themselves around little circular proteins. And I'll draw them out for you. Just, I'm really picky about the colors. So there is like a circular protein, and the DNA is basically going to go and wrap themselves around these proteins. Okay, and they wrap themselves around it, and wrap themselves around it. There you go. Okay, so these are called histone proteins. And this version of the DNA, where it's all tightly packed around the histone protein, that's called chromatin. Okay, the best way to think about this is you guys know like a yo-yo, right? Assume you have played a yo-yo before, or your kids have played a yo-yo before. Okay, the rope of the yo-yo is like the DNA, and the yo-yo thing itself, it's like the histone proteins. So you can take the rope and you can wrap it around the yo-yo, right? So instead of having like a long rope that gets tangled easily, now they're all wrapped around the yo-yo and it's nicely packaged. Does that make sense? The only thing that's a little bit different between the analogy of the yo-yo and what's actually happening is like there's not just one yo-yo because you have a lot of rope. So after you finish wrapping around one, you gotta loop it on the other one, and then you loop it on the third one. And in the end, you get this whole collection of protein and DNA packaged together. That is what we call chromatin, and we will fit those into our nucleus. Any questions so far? Okay, so each cell has approximately like 2 meters of DNA. Right? 2 meters of DNA all packaged into the nucleus. So you might wonder, how is this different from chromosome then? Well, it turns out if you take the chromatin and then you package it even further, okay, with more packaging protein, okay, then you will get your chromosome. Okay, and sometimes chromosomes, they can make a copy of themselves. Okay, and then you would end up having like two of these things stuck together. That's why, like on TV, if you see someone looking at chromosome, it's usually like a X pattern. But that's what it is. It's basically the same thing, but even more compact. So chromatin and chromosome are essentially the same thing, but chromosomes are more condensed. Okay? And you only need to change it to chromosome when you are moving them around in the cell, okay? Such as during cell division. Okay. Sorry, there you go. Is everybody okay with the DNA, chromatin, and chromosome? Okay, I hope so. Thank you. So let's go back here. So there, this is basically the nucleus and all the features associated with it. Now let me help you understand this a little bit better. You don't have to draw this next part. I'm just gonna draw it to help you understand better. If you need a bit of clarification, you can think of this as like an affogato, okay? So there's your affogato seed in the middle, and then you're gonna have the rest of the affogato here, and of course the affogato is going to have some skin as well. Okay, so in this analogy, the seed of the affogato would be like which structure? Like if the whole thing, yeah, sure, go ahead.
Nucleus.
Oh, yes, I guess I wasn't being clear. Yeah, it's my fault. The whole thing is the nucleus. Okay, let's say the whole thing is the nucleus. Then the seed would be like the nucleolus, right? Yeah, this,
Nucleus.
yeah, nucleolus. And then the meat would be where you will find the chromatin. And now the skin of the avocado, that would be like the nuclear envelope. Okay, do you guys see it? If you take a toothpick and then you poke a bunch of holes on the skin, then those would be like the nuclear pores. Okay, I want you to be able to visualize this so it helps you understand a little bit more. Okay, so like if this whole thing was the nucleus, and then you poke some holes on it, right? Those would be like the nuclear pores, and so on and so forth. Are we okay for the 5th time? Sorry, I'm a little bit out of it today. Not feeling too well, but hopefully you're still able to follow. Okay, so that's that. Now, this is what we're gonna do next. Right? The nuclear envelope that we just drew, the purple stuff, it turns out the nuclear envelope does not end around the nucleus. Okay, the nuclear envelope will extend outward from the nuclear envelope, and it will form these networks of membranes. Okay, they can be very, very long, like that. So these here is continuous with the nuclear envelope. Right? It extends into the cytoplasm. And we have a name for it. Okay. Actually, there are two kinds. Okay, I'll draw another one here. Okay, there are more that will extend out here. Like that, use a different color if you have. Okay. So some of these networks, they are going to have ribosomes attached onto them. Okay, I haven't told you what they do yet, but the ribosomes, we know they came from the nucleolus. Okay, and then there are going to be the same kind of network, but without the ribosomes. Okay. So the ones with the ribosomes on them, the name is rough endoplasmic reticulum. Okay, the short form is RER. Okay, they are covered with ribosomes. Now, on the other hand, these guys, it's the same continuation of the network, but they do not have the ribosomes. And when you don't have the ribosomes and you look at them under the microscope, they appear to be smooth. So it's called smooth endoplasmic reticulum. Okay, or SER. So copy what you need to, and then I'll tell you more about it. So now will be a good time to tell you more about the ribosomes. Okay, some ribosomes are attached to the endoplasmic reticulum, but other ribosomes are actually free floating, and they just exist on their own. Okay, so you can just put some dots here and there. So these are free floating ribosomes. If you zoom in onto the ribosome, like here, if you zoom in onto it, you will see that the ribosome actually is like a hamburger. Okay. There is a top subunit, and then there is a bottom subunit. And the job of the ribosome is to synthesize proteins. Okay. So they are really small, but if you zoom in, it looks like a burger. There's a top bun and a bottom bun, and they work together to make proteins. You remember proteins, right, from last class? They are made up of amino acids, and you need to connect the amino acids together by creating the peptide bond. And so the ribosomes, that's that for you. It will find the right amino acid, and it will create the bond and chain the whole protein together. So they could do it by themselves, floating around in the cytoplasm, or they could partner up with the rough ER and synthesize proteins together. Okay, so the free floating ribosome can make protein, and here the rough ER covers with ribosomes, and they can synthesize and package proteins. I'll show you how that looks like in a bit. So let's take a closer look at the rough ER. All right, so let's zoom in again. There's a lot of zooming in going on here in this picture. Okay. Let me redraw it for you. Okay, so this is like one of these things, but like zooming really close. The inside is actually a hollow, and here we're gonna have the ribosome on it. Okay, so I'm gonna draw the ribosome, the hamburger. This is ribosome. And the ribosome is building the protein. Okay, so here's amino acid, amino acid, amino acid. And it's going to be building the protein right in the inside of the, oops, totally erased it, in the inside of this rough ER. Now, once you finish making the protein, then the protein is going to fold up into a ball, right? You know, like the tertiary structure. And this protein is going to move towards the edge. And eventually, and this is really hard to show on a static picture, but please try to visualize it. And if you don't understand, ask me. What happens is this thing will actually push itself towards the edge. It's like blowing a soap bubble, and eventually it will bubble off. Sorry, are you guys still there? I think something weird with Zoom going on here. Can you hear me?
Yeah, we're still here.
Okay.
Yeah, we can hear.
Sorry, I just got like a pop-up from Zoom saying it's like, anyway, sorry. Okay, so this is gonna go and bubble off from the edge, and eventually it will completely detach, and then it will have the protein inside. Okay, it will have the protein inside. All right, so let me make some quick notes here for you. Okay. Ribosome synthesize the protein in the rough ER, which gets packaged into a vesicle. Okay, so this vesicle is going to bubble off from the rough ER. Now, I just want to reiterate, the ribosome doesn't have to work with the rough ER necessarily, okay? It can do it on its own, but if it's producing protein on its own in the cytoplasm, then those are not going to get packaged. Okay. All right, I think we're going to take a quick break here. We'll resume in about 5 minutes. Let's come back at 1:33, and we'll write a little bit more notes. Okay, see you shortly.
What did you write in the last line? Which gets packaged into it?
Vesicle,
Oh,
VESICLE. Yeah.
Oh, vesicle.
Okay, 1:33. We'll come back. All right, let's keep going, guys. So that's the rough ER right there. It's primarily responsible for protein synthesis. Okay, but ribosome is the one that's making it, and the rough ER, it's sort of like packaging it into these vesicles. We're going to take a look at what it does in a bit. Okay. Now, just a quick word about the smooth ER, and then I think we're going to switch back to the PowerPoint for a second. Okay. For the smooth ER, which again is an extension of the nuclear envelope, they are different from the rough ER in that they do not have ribosomes on them. So do not have any ribosomes. And the main function of the smooth ER is to synthesize lipids. Okay, so what kind of lipids do we need to make in the body? Do you guys remember different types of lipids? Give me like 2 examples of lipids that you remember from last week, perhaps.
Like wax, everything that is oily.
Right. So give me something specific, like that we make in the body, like for like some kind of lipids that we need. Anything besides wax? Phospholipids, yeah. So if you need to make more phospholipid, smooth ER is the place to do it. If you need to make steroids, right? The
Steroids, yeah.
testosterone, yeah, and the estrogen, you will need to make it at the smooth ER. Okay. They will also help break down lipids and help detoxify drugs. Okay. So if you're taking medication, they need to be metabolized. They need to be broken down. And the smooth ER will have a role in doing that. Okay. Okay, let's go back to the PowerPoint slides. So this is like an overview of all the stuff that is to come, and so I'm not gonna spend too much time on each slide. They pretty much cover the same information, so you can read through them yourself later on. But here we go. This is the nucleus, and then in the middle we have the nucleolus. Okay. What does the nucleolus make again? Do you remember? Look at the diagram you drew. What does the nucleolus make? Okay, if you don't remember, that's okay. Here is the nucleolus, and it makes ribosomes. Okay, it makes ribosomes. Okay, and you can see here the nucleus is surrounded by the nuclear envelope, and you see all the pores here. These are the nuclear pores. Right? So when the nucleolus makes the ribosome can exit the nucleus through the nuclear pores. In this diagram, the yellow dots are the ribosomes. This talks about the DNA and the chromatin and the chromosome. We already explained that. This talks about the nucleolus in the middle and how it makes ribosome. And you can see the ribosomes are made up of a large and a small subunit. They are typically apart. They only come together when they are making proteins. Right? So remember, some of the ribosomes they are free floating. Others are attached to the ER. The ribosome themselves are made up of protein and RNA. So ribosome makes protein, but it itself is also made up of proteins and RNA. In this picture, you can see these are like free ribosome. And then there are some that are attached to the ER. Attached to the ER, which makes them rough ER. I'm just going to read this to you. Ribosomes are made in the nucleolus and exits into the cytoplasm through the nuclear pores, the opening that are found on the envelope. And we have been saying this a couple times already, but some exist freely in the cytoplasm, while others form clusters and attach to the endoplasmic reticulum. The functions are the same. They're both going to be making proteins, but if it's free by themselves, then the proteins are not packaged into vesicles. If it's with the ER, then you can put it in a bubble. We call the bubble a vesicle. Okay, so we should talk about this central dogma of biology. Okay? Dogma means theme. Okay, central theme of biology. By now, you should know DNA contains information to make proteins. But DNA are found inside the nucleus. We don't want to damage the DNA because, if you damage the DNA, we learned last time, then the instruction might be different for making the protein, and then you might end up creating non-functional proteins, right? So we don't want to damage the DNA. We want to keep the DNA in the nucleus where it is safe. But we want to access the information from a DNA and use it to make a protein, right? So think of it as like this. At the library, there is this, master cookbook. It has all the recipes in it. Right? They don't let you sign it out, right? But you really want to use a recipe and make it at home. So what do you do? Okay? What can you do? You can't take the book out, but you want a few pages. So what do you do? Well, you're probably just gonna take a picture with your cell phone, right? Okay. But before there were cell phones, you will copy, you will make a photocopy, right? And then you can take that copy home and then use it to make the whatever, right? The lasagna or something like that. Okay. And that's basically what happens here. We have the DNA, which is the cookbook, in the nucleus, and we cannot take it out because it's too big and it's too dangerous to take it out. We might damage it. So we make a copy of it. So the copy of a DNA that we make is called a messenger RNA. Okay? This copy leaves the nucleus through the pores. And then we're going to use that copy. The ribosome is going to use that copy to make the proteins. So this process from DNA, and you make a copy of it in the form of RNA, this is called transcription. Okay? To transcribe is to copy, right? DNA and RNA uses the same language. They are both nucleotides. And then you can use the instruction in the RNA to make the protein. The ribosomes will do that for you. And this is translation, because it's a different language. RNA is nucleotide. What language is protein? What are the building blocks of protein?
Amino acid.
Amino acid, that's right. So you're going from nucleotide to amino acid, which means it's a different language. So that's why it's called translation. And so that's how information flows in a cell. It starts in the DNA, you make a copy in RNA, and then you use it to make protein. This happens in the nucleus, and this happens in the cytoplasm. Okay. We will be revisiting this theme over and over again throughout the course. So we're just introducing it in this particular lecture. Okay, I'm not sure if you guys didn't know the answer to some of the questions I'm asking, or like if you're just like, not willing to say the answer, but please study hard, okay, for the upcoming quiz on Thursday, because you do need to know some of these things. Not the lecture 3 stuff, but then like the things I asked you about, example of lipids or like the building blocks of protein. You gotta know all that stuff for the upcoming quiz. So yeah, study hard. Okay, which of the following is a prokaryote? What do you think?
E. coli.
Yeah. Oh, I was gonna launch the poll, but that's okay. You already said it. It is the E. coli. That is a prokaryote. That is a bacteria, right? Everything else here is a eukaryotic cell. Okay, let's try this one. Okay, the answer is produce ribosomes. We actually wrote it down earlier. That is the function of the nucleolus. Okay, let's briefly talk about the endomembrane system. The endomembrane system, endo means within, right? So basically endomembrane system is a network of membranes inside the cell. Okay? If you drew the picture along with me, we actually have several components in the drawing already that belongs to the endomembrane system. So here they are. The endomembrane system work together to produce, to process, and to digest things like lipids and proteins, okay? And sometimes other large molecules as well. And there are some parts of the endomembrane system that will help recycle some of the resources so we can use it to build other kinds of molecules. The endomembrane system include the following structures. There's the nuclear envelope, there's a rough ER, the smooth ER. These three things are already in your picture. We will talk about the Golgi and the lysosome shortly. Let's do a quick revision of things that we already know from the drawing. The rough ER. Right? In this picture, this portion is the rough ER. It's an extension from the nuclear envelope, and it's covered with ribosomes. Okay? We said it many times already. I'm not going to spend too much time on this. The ribosome will synthesize the protein directly into the ER. This was what I was trying to draw with you. We have a ribosome here, and you can see the protein is being synthesized right into the inside of the rough ER. The protein that moves towards the edge where it will bubble off, it will butt off into a vesicle. Okay, so the protein inside the vesicle has several places it can go. Right? And we're going to look at that in a second. The smooth ER, on the other hand, right here, has no ribosome stuck on them. And they are important for making different types of lipids. So for example, steroid hormones like progesterone, estrogen, testosterone, they are produced by the smooth ER. Now you got to understand, every cell in the body does not necessarily have the same amount of each organelles. Okay? Cells that are specialized in certain functions might have more of one type than others. So in the ovaries or the testicles, the cells, because they need to make these steroid hormones, you would expect to find more smooth ER in them. Okay? In a cell that is specialized in protein synthesis, then you would expect to find more rough ER in them. The smooth ER, as we said, is also useful for breaking down fats and detoxifying drugs. So the liver, as you may know, is an organ that's responsible for cleansing our blood, detoxifying the blood, and you would expect to find more smooth ER in the liver when the body is exposed to drugs. And when I say drugs, it's not necessarily like illegal drugs. Like when you take medication that helps you, those drugs needs to be metabolized as well, and that typically happens in the liver as well. All right, so now there is another thing called the Golgi body. I'm just gonna add that to our note here. Okay. So this is the rough ER, and by now you should understand the rough ER is basically going to be churning out these vesicles. Okay, and the vesicle has proteins in it. So this guy here is a vesicle from rough ER. And where it is gonna go is to the Golgi body. The Golgi body is several layers of membrane kind of stacked together like that, like a pancake, okay? Several layers of pancake. And the vesicle that's coming from the rough ER, they will fuse with the first layer, and then the contents actually get passed forward and sometimes backwards. It goes back and forth a couple times before it's repackaged into a new vesicle. Right? And the protein inside by that point is usually modified. And that is the function of the Golgi body. I'll write down for you, Golgi body. Sometimes it is called Golgi apparatus. And the function of the Golgi is to receive the vesicle from the rough ER, and then it will sort, modify, and repackage the protein. Okay, it's like the shipping center of the post office. It gets all these packages from all over the place. They gotta sort them. Sometimes they need to modify it. Okay, maybe there's some damage. They're gonna modify and repackage it before shipping them off. Right, and that's basically what the Golgi body does. They will modify the proteins in several ways. Sometimes you need to add things to the protein. Sometimes you need to cut the protein. Sometimes you need to, do other things to the protein. Lots of things you can do to it. We're not going to go into the details. And once you've done modifying them, the Golgi body will repackage it in a new vesicle. Okay, so this is a new vesicle with modified protein. Okay. Now this vesicle can go a few places. Yes, this will be posted. Okay. So if you can't copy everything just now, it's okay. Right? You can add to it later. And all this is in a PowerPoint, anyways. Okay. I just like drawing it out with people because I think it helps them understand better. Okay. Yeah, you're running out of space to draw. I know. Sorry. Okay, we're almost done anyways. So look, listen, right? We are looking into the inside of the cell, and here is actually the cell membrane, okay? I didn't really draw it out earlier, but here is the cell membrane. Sorry. There we go, cell membrane. Okay, so I'm gonna tell you what's gonna happen to this guy, okay, to this vesicle. A couple of things can happen, okay? Number one, these are in no particular order, okay? I'm just writing them down as they come to my mind. Number one, the vesicle can stay in the cytoplasm until the contents are needed. Okay, like maybe you don't need this protein right now. Maybe you need it later. Okay, but you're just making it ahead of time because you never know when you need it. Yeah, it's better to be ready. So you store the protein in the vesicle, you keep it in the cytoplasm, and then you will have it when you need them. That's one place it can go. Another place it can go is actually it can be released out of the cell. Okay, so this part is a little bit hard to draw. Let me try that one more time here. All right, so this bubble is actually going to travel all the way to the cell surface, and it's going to fuse with the cell surface, and in doing so, the protein is going to be released. Okay, so that is a second possibility. Number two, protein is released out of the cell. Okay, and then it might go into the bloodstream for, you know, whatever reason you need it elsewhere in the body. Now, we haven't really talked about this next one yet, but it's sometimes possible for the protein to become part of the membrane. Okay, we know the cell membrane is made up of phospholipids and that there are some cholesterol in it, but it turns out there are quite a bit of proteins in it as well. So the protein that we just made, they might become part of the cell membrane. Okay. It's also possible for the protein to be you know, in the cytoplasm on its own. Okay? And then you can use the protein for whatever purpose. And then there's one last thing that it can become, and this is the last thing we're going to draw, okay? Number 5, it can become a lysosome. Okay? A lysosome is an other kind of organelle. What color should we use? Let's use, I don't know, this one. Okay, this is a lysosome. And then it has like special protein in it. Okay. Can become a lysosome. So all these things can happen to the protein. I'm not going to write down what the lysosome does because it does a few things. We will go through the PowerPoints for it, okay? Okay, so lysosome, what does it do? The lysosome contains digestive enzymes. We know what enzymes are now, don't we? What kind of macromolecules are enzymes? Do you remember from last week? Please tell me you do. Are they carbohydrates? Are they protein? Are they lipids? Are they nucleic acid? What are
Protein.
enzymes? They are proteins, that's right. Okay, and they are proteins that helps speed up chemical reaction. In this case, the enzymes are going to help you digest things. So inside the lysosome, there are different types of digestive proteins, and it will help break down macromolecules. So if the cell ingested some kind of food, then it will merge with the lysosome, and the enzyme will help digest the food. Okay, your white cells can do that. They might eat up a bacteria or something. Then you need to digest the bacteria. Sometimes organelles might get too old or damaged, and so we need to destroy the organelles, free up some resources. Lysosome is going to do that for you. Okay, so don't forget the lysosome came from this whole process, right? And inside the vesicles are proteins. So when it becomes a lysosome, the enzymes inside, they are actually proteins. Sometimes the lysosome can release its enzyme and cause the cell to die, okay, deliberately. So this process is called apoptosis. Apoptosis is programmed cell death. Okay, programmed cell death. Sometimes you need to kill off a cell. Maybe the cell is sick, or maybe it's part of normal development. When we were growing inside as a baby, inside of our mothers, there are actually web between our fingers, right? And you can see like a little bit of web here, right? Here. And the cells that forms the web needs to die off so that our fingers can be free. How do they die? Well, the lysosome is going to release its content, kill off those cells in a process called apoptosis. Okay, there are other reasons why we need to kill off a cell. We will learn that later in the course. Now, inside the lysosome, there are a mixture of enzymes. And these enzymes, as I said, they are proteins. And the instruction to make these proteins are in the DNA. So it's possible for people to inherit mutated DNA, and therefore they are making the wrong enzyme. And depending on which of these enzymes are missing, it could lead to different diseases. Okay, so diseases related to enzymes in the lysosome, in general, they're called lysosomal storage disease. I have two examples here for you. In Pompe disease, the enzymes that are needed to break down glycogen, which are made of carbohydrates, right? It's missing. And so there is an abnormal amount of glycogen building up in muscle, causing it to become weak. And if this happens in the breathing muscle or in the heart, then the person is going to, have quite a bit of complication, right? Another example is Tay-Sachs disease. This is one of the things people can test for in babies before they're born. Tay-Sachs disease is where you're not able to have lipid digesting enzymes, and these lipids built up in nerve cells in the brain, interfering with normal brain development. Okay, so there's a whole list of them. If you Google lysosomal storage diseases, like there are at least like 20 to 30 of them that are related to these enzymes. Okay, ribosomes are made up of, there are two things that makes up a ribosome. Let's see if you remember what they are. That's right, they are made up of RNA and proteins. Yeah, I only have two people who answered it, but those are the correct answer. Okay, which of the following organelles are involved in breaking down macromolecules, toxin, and wastes? Let's try this one. There are more than one correct answers here. Okay, many of you chose lysosome, which would be correct, but the smooth ER also help breaks down toxin, right? Yeah. So these two things are the correct answers. All right, I think that's where I'm gonna stop with the with this lecture. We'll finish this thing up late on Thursday. Now, before you go though, I did promise you I'm gonna take up some of these questions from lecture 2. These were concept check questions that we didn't have time to finish last time. So I'm just gonna go through the answers with you, okay? I'll give you a moment to take them out if you need to. All right, first one, a peptide bond forms between which group? Now, if you remember, the amino acid look like this. And you basically, if you have two of these things together, you need to connect this group with this group together. Okay. In the notes, we wrote it down. This group here with the COOH, that's called a carboxyl group. And this here is called an amino group. So to form a peptide bond, we need to connect those two things together. Carboxyl and amino. Hemoglobin is an important protein that is composed of 4 polypeptide chains arranged in 3D shapes. What level of protein structure is illustrated by hemoglobin's 4 polypeptide chain? Whenever you have multiple polypeptide coming together to form a complex, then it has quaternary structure. Quaternary structure. Pancreatic cells make insulin, which is a type of protein. These cells use something to synthesize insulin by the process of something. Okay, so it doesn't matter what pancreatic cells are, even if you don't know what it is okay. And even if you don't know what insulin is, that's okay as well, because the question tells you it is a type of protein. So basically, the question is asking you, how do you build proteins, right? So the monomers for proteins are amino acid, and to connect amino acids together, you need to do dehydration synthesis. Dehydration synthesis. Which statement about proteins is false? Okay, let's take a look. The primary structure of protein is determined by the sequence of amino acid. That's correct. If you read the amino acids from one side to the other end of the protein, that is the primary structure. It is, of course, determined by your DNA. Example of secondary structure include alpha helix and beta pleated sheet. That is also correct. Tertiary structure is stabilized by hydrogen bonds of the peptide backbone. That is not true. A tertiary structure is stabilized by interaction of the side chains of the R group. The last one is also true. We wrote it down. Increasing temperature will disrupt the secondary, tertiary, and even the quaternary structure, but the primary structure is going to remain unaffected because the covalent bond is too strong for it to be disrupted by heat. Okay, so this is the false one. That's the one we're going to choose. Any questions? Okay, we have a few more here at the end. Which of the following statement is true regarding purines? Okay. Does anybody remember what purines are? There are two nitrogenous bases that are purines. What are they? Out of the ACTGU stuff, which ones are the purines?
A and G.
Very good, A and G. With that in mind, what's that?
Purines.
That's right, the purines, that's right. So with that in mind, let's take a look at the answers. Adenines bonding to thymine is stronger than guanines bonding to cytosine. That's not true. I think I might have forgot to mention this last time, but between A and T, there are only 2 hydrogen bonds holding it together. Between C and G, there are actually 3 hydrogen bonds holding them together. What that means is if you have a strand of DNA that has a lot of C's and G's in it, it's going to take more energy to separate them. On the other hand, if you have a strand of DNA that has a lot of A's and T's in it, like 60% A's and T's, that's going to be easier to pull apart compared to something that has like 60% C and G's in it. Now, it's important to know this, like in real life application. You might heard of like PCR tests, right? During the pandemic, you might need to do a PCR test to see if you have COVID or not, right? Okay, so PCR actually involves separating DNA, as we will learn. So it's important to understand the GC and the AT contents in the DNA in order to know what temperature to run your reaction in. Okay, so we'll learn about this later on in the course. But the key thing to remember here is only 2 hydrogen bonds holding A's and T's, 3 hydrogen bonds holding G and C. So in this case, A is not correct because A and T are actually weaker, only 2 bonds, weaker. Cytosine is a purine. No, it's not. Okay, only A and G's are. Purines bind readily to deoxyribose, but not to ribose. Deoxyribose are found in DNA. Ribose are found in RNA. So the purines, the A and Gs, do they only bind to DNA and not RNA? It's not true. Okay. A and G are found in both DNA and RNA. So they would bind equally well to each. Both adenine and thymine are purine. Nope. Thymine is not a purine. Purines have double-ringed structure. That's the correct one. 2 rings for purines, 1 ring for pyrimidine. Couple more. Which of the following is not a feature of DNA? Select all correct answers. Nucleotides within a single strand are connected by covalent bonds. That's true. Okay. All the macromolecules we learn are connected by covalent bonds. Adenine form base pair with uracil. That is not a feature of DNA because there are no uracil in DNA. There are only thymine. Complementary strands, that means each strands, are held together by hydrogen bonds between, as I show you in the diagram, they are held together by the nitrogen base and not the phosphate group. The phosphate group makes up the backbone. And the last one is true, right? 2 characteristics of DNA is that it's complementary, you can predict the other strand, and that they're anti-parallel, right? They run in opposite direction. And this is the last one. A chemical reaction occurs slowly at 37 degrees. When molecule X is added, the reaction speeds up significantly. What kind of molecule can speed up a chemical reaction? Everything we learned, which one does that?
Enzyme?
And what kind of macromolecule is an enzyme? Like, is it a carbohydrate, protein, nucleic acid, lipid?
Protein.
What is it? Yeah, so it's a protein, right? So this is the first thing you have to realize, that molecule X is a protein. Okay? And then the question is asking, which of the following is a monomer of molecule X? Right? We all know a monomer of a protein is an amino acid, and that's what an amino acid looks like. Okay, this here is a nucleotide. This is a sugar, and this is a fatty acid. So the answer is A. All right, don't forget we do have class on Thursday, but you also have to do your quiz, which is available to you from 6 to 11:59 p.m. Make sure you study for it before you attempt the quiz. There will only be 1 attempt, and it's worth 10% of your final grade. If you have any other questions before writing the quiz, feel free to email me. I won't be able to answer any questions on the day of the quiz, okay? If you need to ask me things, it has to be tomorrow, okay? Also, there is the tutor available to you. I don't know if I mentioned that already. I posted an announcement about it. It's on eCentennial. Maybe I can quickly show you. For those of you who did not see it, your tuition actually covers complimentary tutoring service from the library. And this semester, we have a co-op student. His name is Kian. And you can book an appointment by clicking on this link. There's a form you can fill in. And he's available on most all days, every day, basically, sometimes in person as well if you need that kind of help. These things do get booked up really quickly, especially closer to test dates, so make sure you secure your slot earlier rather than later. Okay, I am also available for extra help if you need it. Just send me an email. We'll figure something out. Okay, thank you guys. Take it easy, have a good day. Bye for now.